I'm actually a Minnesota native, so I grew up as a Gophers fan. So they're already bowl eligible this year, so that's satisfactory for most Minnesotans. But I will be rooting for the Gophers going forward here. So there's not a whole lot I can really boast about the Gophers for too much. You got to cheer for them when you're from Minnesota. So as I said, I went to Iowa State. Actually, did a PhD in biochemistry and immunology while I was there. So yes, I'm going to talk about in utero exposure to choline and the long-term effects that has. And I really want to recognize Charlie Staples. I think many of you may know Dr. Charlie Staples. He was at the University of Florida for I think around 30 years. He spent his career, his whole faculty career at the University of Florida. And he is really the one that took on this work, developed this work. As a new faculty member, I was just fortunate that he kind of got me plugged into this and working on it as well. And the work with choline that he is doing there. So he is, to many people, a friend, a mentor, a scientist, a dairy scientist, and really this, he should be the one here that's really presenting to this because it was the work that he was doing. And unfortunately, he lost his battle to cancer back in June. So moving on to the what we've been talking about here, and you hear this term fetal programming thrown out there quite a bit in epigenetics. What is that? So that's what we're going to kind of get into a little bit here at the beginning before we get into what's the role of what actually response do we see to this prenatal exposure to choline. So most of you probably may recognize this cow over here. 3918, I believe, is her number, the world record for milk production. So she has this phenomenal phenotype. It's just amazing what that cow can do. So obviously she has a genotype that supports that. She has this genetic potential to be able to do that. But it's not just her genes alone. So there's roughly 30,000 genes that this cow has, somewhere around there. From there, about 100,000 proteins, approximately, that would be produced from those genes. So to get that phenotype from her genotype, there's an interaction with the environment that takes place. And so this is going to include some things that we can control very well, such as her nutrients. Some things that we may not be able to control as well, sometimes pathogens that if we have good practices, yes, we can control those, but some of those we can't always control. Her microbiome from her gut microbiome would be the most important here, I'd say, but in general, her microbiome. And then several stressors. These are just examples of her environment collectively that will interact with her genotype to result in that ultimate phenotype that she has. And so this interaction really begins before she's born. It begins from the point of conception. In fact, really even before that, there's maternal and paternal imprinting that actually take place before that. But from what we can start to control, it's from the time of conception. And so you take any given cell in this the cow's body, and it's going to have all her genetic material. And this is what's amazing to me is every cell in the body has, for the most part, the same genetic material. Some minor exceptions to that, but it's the same, as Isis pointed out earlier, the same code that's in there. The difference, though, is that epigenetic response that alters what gene will be expressed, when and how much and where that gene will be expressed. So obviously a mammary epithelial cell is going to express a different set of genes than, say, the gut epithelium or a muscle cell or an adipose cell. It's going to express a different set of genes, and depending on either intrinsic signals from that, from within the animal or extrinsic signals, determine how much of that gene will be expressed. So in determining that, that's where epigenetics comes into play. And so here, you guys recognize this, this structure here is the double-stranded helix. That's the DNA sequence made up of the 4 nucleotides that Isis pointed out earlier. So it's not just this jumbled pile of DNA, it's packaged into chromosomes, right? You have the chromosomes that put all that DNA together so you can have an efficient mechanism to store and retrieve that information, like a very good library system to be able to store and retrieve that. And so the way that the cell then packages that is in the form of a chromatin, so it winds this around these histone proteins. So think of it as an extension cord. You wrap it up nicely and it's easy to pull out and use the next time that you go and use it. Whereas if you throw it in a pile, it's going to be much harder to use. So my 15-year-old son hasn't figured out that concept yet. I'll wrap it up all really nice and go into the garage and see it all thrown into a heap after he's used it. But that's the concept here of these histone proteins, is they help package that chromatin into an efficient way to store and package, but also retrieve that information when it's needed. So ultimately, when the signal comes about, that particular gene can be expressed. So to facilitate when and where that gene will be expressed, you have these markers. And Isis pointed out some of these earlier. So these, a couple of the very common ones are methyl groups, which that's where choline comes into play, and acetyl groups that come in so they can put tags on those histones, either acetyl or methyl tags on the histone proteins, or methyl tags actually on the DNA itself. So this allows for what I call punctuation, or emphasis on what gene will be expressed and to what extent it will be expressed. So just a general rule of thumb for how this works, the more acetylation you have of these histones in general, and this isn't a hard fast rule, but just in general, you'll get enhanced expression. So more of that gene will be expressed if the acetyl or the histones surround in that DNA region are more acetylated. On the flip of that, if you have a methylation of that DNA itself, you're going to get, in general, silencing of that particular gene. So if you're thinking here, okay, choline provides methyl groups, more methyl means more silencing, that's not necessarily the case. It's when and where. It's the, as Isis pointed out, the potential for methylation. So basically, it's providing the methyl groups there to be there when needed. So you get the proper, say, annotation of this set of DNA when needed. So that's in general kind of what epigenetics is. This, what I say, putting on the punctuation or emphasis on the instructional material that's there. Now, so what is fetal programming? And so I define this as, we're shaping the epigenome via these environmental factors. So some of these we have control over, some we don't have as much control over. So this is where you get interactions between either nutrients, pathogens, microbiome, or the stressors that are helping to shape this the epigenome that's in place there. So this is across the whole genome. So this is a massive amount of material, and we really don't have a good grasp at how all these factors are really influencing this epigenome yet, but we do know it's taking place and that they're critical. We're really at I'd say, still at the beginning, particularly in cattle, understanding really how these different environmental factors are interacting with that epigenome. So I know we have a few Buckeyes in here, sorry Andy, but I need to put this on terms that everybody can understand, because we do have a few Buckeyes in here. So just to put this in an analogy to help everybody understand, because I could talk about this all day because I love biochemistry. So to me it's interesting, but you guys may be saying, oh, it's nonsense. How does this work actually? So as I said, it's punctuation or qualifiers that dictate the meaning or emphasis of the instructions. So take this set of this code right here. If you look at it long enough, you may be able to see what's actually written there. But say some of this here represents the epigenome, where if you include it or take it out, it can change the meaning or emphasis of this statement. So here, from the words that were in there, you can come up with this statement right here: drink almond juice. Well, we have a term in cell biology, if there's too much DNA damage, it goes down this road of programmed cell death. So here we got an outcome of take these instructions, results in programmed cell death. Figured that the dairy farmers would appreciate this one. Well, you actually look at another set of still that same set of instructions is there, of that original whole length that we had there, but then you add in the punctuation or annotation, and here it says, do not drink almond juice, drink milk. So that's another version of what's there. Still, you could take that further. Do not drink almond juice, drink more whole milk every day. So adding more emphasis onto that statement, or changing the meaning completely. And so all that, all that, if you go back here, that's all in there. And so that's kind of what's in place with our genetic material. There's a whole bunch of information within there. But by the use of that epigenome, it's able to provide more clarification or meaning to that, the instruction that's there. So some examples of fetal programming, some that you guys have probably heard of. So a few of these that we've done down in Florida with Jeff Dahl's group down there, looking at the effects of heat stress, and originally looking, okay, what's the effect on production of the cow if she has a late gestation heat stress. Well, they actually found that not only does that affect the cow, it also affects the calf born to that cow. So this is what they found here is in utero heat stress decreases calf body weight to puberty. I'll point out here, this doesn't happen just in Florida. This can happen anywhere that you have heat stress. And certainly in the Midwest, this is going to occur as well. So what they found, when you look here, these calves had decreased weight gain when they were exposed to in utero heat stress. Same thing, so this is even longer term response. If they were exposed to in utero heat stress, in their first lactation then, they had decreased milk production. So an example of fetal programming taking place, it wasn't just a response that affected them early on, this had a lasting response. So these animals had, after they were exposed to that in utero heat stress, they were raised the same way throughout the rest of their life. Same dietary management, same housing afterwards, but yet they had a lower ability to produce milk in their first lactation. So those are a couple of kind of prominent responses that we've seen for examples of fetal programming. So those are one for heat stress. Another one that we did, this is actually in beef cows, were at 230 days of gestation, we gave them an endotoxin challenge. And so then they looked at the heifers that were born to those beef cows at 10 months of age. They went and challenged them again with doing an endotoxin challenge. They used LPS from E. coli. And here's, they measured IL-6, so an inflammatory marker that they measured in serum. And we see here, these heifers at 10 months of age, when they had been exposed to this either no LPS or LPS in utero, they had a difference in their response to or the response to that endotoxin challenge later in life. So here's an example of affecting the immune system. Another one, this is the same experiment. We looked at the neutrophils, the leukocytes, and one of the genes they express is an antimicrobial peptide. And so these are beta defensins, and we see here there's several of these that are affected by that in utero exposure to this endotoxin challenge. So another example of how early life events, before that animal is born, can result in later life differences in their ability to either, in the case of heat stress, grow, or here their immune response. So this is where we bring in choline. So the evidence that we've seen lately, and certainly Isa's pointed out, this has been known for human nutrition and the models that they've used with rodents. Late gestation exposure to choline is critical for heifer development, and that's what we've been finding out from the work that we've been doing at the University of Florida. And a couple reasons why this may be. Okay, certainly that choline is providing those methyl groups. Also, here's some data from the human gestation and the fetal development. And so phosphatidylcholine, part of that cell membrane, is greatly increasing here in late gestation. So it's very critical for the development of that fetus. So the first bit of work I'm going to talk about here is actually the objective of this experiment was to look at the effects of late gestation rumen-protected choline on in combination with prepartum caloric intake on milk production of those animals and development of fatty liver and energy metabolism. So here's the experimental design. In this one we had 4 dietary treatments where they either had excess energy intake prepartum or maintenance energy intake prepartum, and then they're supplemented either no reassure or 60 grams a day per reassure. So they started feeding this during the dry period at 47 days. They started the prepartum when they started the the high energy diets, and then choline, the rumen-protected choline, is top dressed for the last 21 days prepartum to 21 days postpartum. So, like we've shown in several studies, and this has been shown in a meta-analysis as well, feeding rumen-protected choline during this period improves milk production of these cows. That's something that's been pretty well established now, that you're getting improved milk production from feeding choline during this period. But the surprising observation we had from this experiment was when we went and looked at the heifers born to those cows in that experiment. And so it wasn't one of the objectives going into the experiment. Marcos and Charlie said, hey, let's look at the performance of those heifers. And so it's fairly small in terms of the number of animals here in this first one. But here you just minor differences in body weight. They're actually slightly lighter in the calves born to choline supplemented cows. But then you look at 12 months of age. We had increased weight gain at that point. Post-calving, after those heifers had their first calf, they again increased weight gain. So our average daily gain from weaning to yearlings increased from 0.85 kilograms per day to 0.89 kilograms per day. So this is, again, it wasn't the primary objective of this experiment to look at the long-term effects of late gestation choline, but it's something that we observed in that experiment. So then we went on to design another experiment to more specifically test that effective late gestation choline. And the other part of this experiment was not only look at the in utero exposure, but also the effect of the colostrum, because we saw in that experiment the cows that were fed choline had increased colostrum IgG, both in the concentration and the total amount of IgG that those cows produce. So the hypothesis is the heifers born from dams fed ruminally protected choline during late gestation, they would produce produce colostrum that would have improved quality, and then also it improved both the immunity, health, and overall growth of those heifers that were born. So then we designed an experiment to test that hypothesis. So in this experiment, we had, again, 4 treatments. In this case, though, we went either no prenatal choline, or the cows were given choline, rumen-protected choline in the last 21 days of gestation. And then from that, the calves that were born to those cows, they either received colostrum from a control cow or a cow that was fed rumen-protected choline. So then we could piece apart whether or not it was an in utero exposure or the effect of the colostrum. So we had 4 treatments that the calf ended up being exposed to here. So throughout you'll see this NN refers to it was not exposed to choline at any point, either through in utero or colostrum. The NC here is going to be exposed to, or not exposed to choline in utero, but exposed to choline from the colostrum. And vice versa. The CN here be exposed to choline in utero, but not colostrum. And then here, choline both in utero and in colostrum. So we had 110 cows, I believe, enrolled in this experiment. So about 50-50 for bulls and heifers. I'm going to present some of this data together for the bulls and heifers, but then we did different things with the bulls and heifers, so we won't be able to look at all the data together. So I'm going to touch on some of the heifer data and then some of the bull data that we have. For the prepartum diet, here's the information on that. Had methionine added. We had rumen-protected choline in the form of Reassure at 60 grams per day for the last 21 days of gestation. And then for management of those calves, we fed them the same amount of colostrum and made sure that it had at least a Brick score of 22 or greater. And then they got 2.8 liters of milk replacer twice daily, and then ad litem, starter grain, and water. Just to give you an idea of the setup that we had there, so in a freestall barn that the calves were born, individually housed for the calves from birth to weaning, or from the bull calves from birth till they were sold. So here's the first, this is just survival data, and again, this experiment with only, we had just over 100 calves, I think 107 total calves in this experiment. So fairly minimal to really look at survival or morbidity data, and we only look at it out to 24 days of age here, and that's because the bull calves were enrolled in an LPS challenge at that point. But just to give you an extreme here of what's going on. So this would be a representative of an animal not being exposed to any choline, either in utero or from colostrum. And here would be the calves exposed to choline both in utero and colostrum. And so this would be practically on farm. Here's the difference, because if you're feeding rumen-protected choline in late gestation, you should be having all your animals that you have both in colostrum and in utero exposure. Now I'm admittedly a little bit embarrassed to show some of this data here, because this shows how bad we were at our calf program at this particular time. So this is definitely extreme, but this is maybe unfortunate for our dairy, but maybe fortunate for Balchem. We hit a real stressful period there. We had a really bad time in calf morbidity and calf loss. But here it shows the drastic difference that we saw in this. So that's just the survival data. What did we see? So I mentioned there's an impact on colostrum quality. And here, when you look at, so this would be the apparent efficiency of absorption for IgG. So here's calves not exposed to choline and colostrum in these two bars. And then here, these are the calves exposed to choline and colostrum. So we had an increase in apparent efficiency of IgG if there was if the choline or the colostrum was from a dam-fed choline. You'll notice here actual intake of IgG, or what those calves were offered, did not differ. So it wasn't just a matter of the amount of IgG that was there. There's another factor in that colostrum quality that actually affected how much of that IgG was going to be absorbed. I should point out this is just, in this case, the measurements from the heifers, and we had 59 heifers in that case. When we look at the growth of these heifers, similar to what we saw in that first experiment where we just observed the difference from weaning to 12 months of age, no difference at birth as regards to a statistical difference. 56 days of age, so at weaning, no difference in body weight. But then we look at almost a year of age, we see a difference there again of the heifers that were exposed to in utero choline had a greater average daily gain from weaning to 12 months or almost 12 months of age. Yes, 10 months of age in this case. When we look at intake of starter by these calves, so this is a proportion of calves, heifer calves that consumed 1.4 kilograms of starter, and the day that they achieved that amount, so that for 3 consecutive days. So as Dr. Hutchins pointed out earlier, getting those calves on starter as soon as possible is something we want to try to achieve. So here are the calves that were not exposed to choline either from colostrum or in utero. So you can see they had a delayed time to when they started consuming that amount of 1.4 kilograms of starter. Here we look at milk replacer intake and starter intake. So this line here, this is their milk replacer intake, and here's their starter intake. So this is just from the in utero effect from exposure to choline. So the blue line here is no exposure to in utero choline. The orange line is exposure to in utero choline. And you can see here within this first couple weeks here, these calves had greater intake of milk replacer. And for those of you involved in raising calves, you know this is a time when they have increased incidence of scours during this period too. And when you look at the fecal scores from those calves during that time, that's when that kind of peaks right around this week to 2 weeks of age. When we look at starter intake, here we have the calves that were exposed to in utero choline had increased starter intake faster within this first 21 days of age. The effect of colostrum was also there. So when you look at, this is again starter intake, but this is looking at the colostrum effect. So the calves that were exposed to or fed colostrum from dams that had received rumen-protected choline had starter intake, greater starter intake there within 21 days of age. So the point here is you have both the effect of in utero and the effect of colostrum that are benefiting these calves in terms of their IgG absorption and their milk replacer and starter intake early in life. So one of those things that really impacts that milk intake, starter intake in those first few days would be whether or not those calves get sick. And so when you look at the incidence of fever, and this again is looking at the here the either no in utero exposure or versus in utero exposure. So we had lower incidence of fever of those calves that were exposed to choline in utero. And this would have been of 59 calves here, heifer calves. So then that was mostly data from the heifers. So from the bull calves that were born to those cows, we did an LPS challenge on those at about 21 days of age, really to stimulate this very acute endotoxin type of response. So similar to if the calf really got a heavy dose of a pathogen challenge. So something may occur from a viral infection or a pneumonia, where they may have this similar type of a very acute response. Same exact experiment where these calves were born from either the cows exposed to choline in utero or not exposed to choline in utero, and then with or without choline in the colostrum. So what happens in this response to LPS? So what we did here is infused LPS in the jugular vein, and that initiates a very rapid inflammatory response where these calves will produce these couple of the key pro-inflammatory cytokines here, TNF-alpha and IL-6. This stimulates acute phase proteins from the liver, stimulates protein and energy metabolism from the muscle and adipose. And then you also get an increased body temperature here, the febrile response that will occur. So in this experiment, we had 52 bull calves that were used. And then so here's an example of putting a catheter in those calves. And then we collected blood samples almost every hour out to 8 to 10 hours there. And then all the way out to 72 hours, and we collected that last blood sample to look at this acute phase response. So what did we see in this? Overall, I'll sum it up as there's I call it increased tolerance to this endotoxin challenge. So here this is looking at, I think this got cut off here, but this should be rectal temperatures of these calves. This should be 39.83 to 39.8 degrees Celsius. And you can see here the calves that did not receive choline in utero had a greater rectal temperatures in the early hours after this LPS challenge compared with the calves that were exposed to choline in utero. So a couple of the other physiological responses that go along with that, the respiratory rate, so their breaths per minute, went right along with this increased rectal temperature. Same thing with the heart rate. So the calves that were exposed to choline in utero had what I call an increased tolerance to this endotoxin challenge. So a few of the serological responses, and I should have this one memorized. I think this one here should be TNF-alpha, I believe is what this one's measuring right here. And then we have serum amyloid A and haptoglobin and cortisol. So each one of these shows this effective in utero exposure to choline, where those calves showing the same pattern as the rectal temperatures, the calves that were exposed to choline in utero had a less severe response. So I think of this in terms of, hey, the calf gets exposed to a pathogen that's going to cause pneumonia or some other viral disease that will cause this very acute challenge. They're going to be able to weather that storm much better compared with the calf that was not exposed to choline in utero. A couple other, so this is some indicators of metabolism, some glucose, fatty acids, BHB, and blood urea nitrogen. So typically each of these will go up in response to particularly like the fatty acids and BHB. They will really increase in response to this LPS challenge, what we normally see. And here you can see for the case of fatty acids and BHB, they're not going up as drastically. And then the flip of that, glucose, which will normally drop, in this case, you don't see it drop as much for the calves that were exposed to choline in utero. So, as you'd expect, if they're not having a severe response, they're also going to have increased feed intake. So these calves that were exposed to choline here, they had, after this LPS challenge, they had increased feed intake, or starter grain intake during that time. So again, showing that they're able to weather that storm of that acute challenge that we gave them much better than the calves that were not exposed to in utero choline. And so then when you look at, here's the body weight of these calves after we gave them that challenge. Again, here 2 weeks after that LPS challenge, those calves that had received in utero choline exposure, they had greater body weights after that time. Again, showing kind of the end result of what you'd expect there from that. They're able to weather that storm even better. So what are the key take-homes from this then? Calves exposed to choline in utero during the last 21 days of gestation. Again, that's the time that we fed this. So that doesn't take, that's a fairly short period of time when you look at the whole spectrum. And this is regardless of colostrum source, they had an enhanced ability to deal with challenges prior to weaning. So whether it's the LPS challenge that we gave the bull calves or just natural occurrence from what we saw in the heifer calves, they're able to endure that more. But there's also an effect of choline. Feeding rumen-protected choline in the last 21 days, the colostrum from those dams resulted in greater IgG, apparent efficiency of IgG absorption in the calves that were fed that colostrum. So the bottom line, feeding rumen-protected choline during the last 21 days of gestation, in this case is 60 grams per day of ReaShure, has positive benefits in the offspring via in utero exposure and the benefits of the colostrum. That's the bottom line here. So not only do you get that benefit in the cow from the milk that she's producing, you're also really benefiting the calf here, and that's a long-term development that's taking place there. To achieve those goals in heifer development that Dr. Hutchins was talking about earlier. So with that, there's, again, big thanks to Dr. Charlie Staples. He's really the one that designed this work, took it on. Marcos Zanobi, who is also with Balchem, but in the other southern hemisphere now. He is the one that, the graduate student that really did this work too, and many others that helped along with that. So with that, any questions? Yes, Isis. Yes, so for why we see this change in immunity there. What we've seen in the cows from rumen-protected choline is there's this dampening of inflammation. I call it dampening. There's many ways you can call it there, but just a dampened immune response, just kind of like what we saw those bull calves. So exactly how that's taking place, is it for sure an epigenetic factor, or is it just something in immune cell development taking place? Being that we see it kind of a prolonged response or delayed response, I'd say there's an epigenetic mechanism there, but exactly how it's doing that yet we don't know. What is there a particular set of genes that it's targeting, or somehow it's programming those cells to develop differently? We don't have an answer to that yet, but based on what you've seen in mice, and not necessarily related to immune function, but we'd say yes, there's an epigenetic component to there. We just don't know how it's doing it yet. Yes, Dr. Kirk.
Did you see a reduced incidence of both respiratory and diarrhea with those cats when they had choline supplement taken before birth?
So when we actually looked at incidence of respiratory and diarrhea, we don't see any differences there. There's only the difference in incidence of fever. So we didn't actually see any difference in respiratory, and I apologize, I didn't have that data put up there in respiratory or scours. There was no difference in those, or even the scores if we quantify the severity of them, but there was a difference in the incidence of fever.
Would you expect to have seen that, though, if it has immunity effect?
Yes, but the number of heifers that we have there, yes, we're limited, and we went into it knowing that we're somewhat limited. Would we truly see a response that way? And fully acknowledging that's a limitation to this is the size of the experiment we have there.
Well,
We'd still have Marcos there rolling cows through if we could, but he wanted to graduate sometime.
You may not have had enough incidents to show a difference. But if you go to a commercial operation, you could probably see it.
Yes, yeah. I'll let Bob talk about that next.
Okay.
Are there any other questions? I have maybe one question.
Yes.
So, the whole field, as I was mentioning during your mic once, sort of exploded. What would be your one takeaway for those of us on the practical side
Yeah, when you look at epigenetics, or when we talk about fetal programming specifically, or all of it together, there's not a point that we can't ignore along the whole pathway. And so we know it happens from conception all the way up. So everything that fetus is being exposed to from conception is going to affect her long-term development. And in fact, it even goes on before that, from maternal imprinting and paternal imprinting, goes on longer than that. But we know, if we focus on those times, we have to pay attention to everything from that point of conception and even all the way up through development of that heifer. That every point along the pathway there is critical. And so we see here, in the case of late gestation nutrition, there's definitely a critical point there, and other environmental exposures that we see. So a key takeaway here, when you look at rumen-protected choline, there's many things that it does. You have the positive benefits for the cow and for that long-term development of that heifer.
Thank you.
All right, thank you.